heredity. Tall gene were represented by capital T and short gene or dwarf gene was represented as small t. So when he crossed the capital T plants with small t plants, means tall plants with purely short plants, the F1 which is produced is capital T small t version. So F1 was not purely tall, but it was tall. That means a single copy of capital T is enough to make the plant tall, while both copies have to be small t for the plant to be short. Traits are like capital T are called dominant traits, while those that behave like small t are called recessive traits. In F1 generation, when the selfing is performed, the capital T small t upon selfing was produced. The selfing in F1 generation produces capital T, capital T small t and small t, and their ratio is 1:2:1. So here Mendel observed only one character. He called this type of cross as monohybrid cross. Now Mendel was interested to see any other characters also in these pea plants. So he observed that there are two types of seeds in these pea plants. One is round seed and one is wrinkled seed. So was there any relationship between round seeds and tallness or wrinkled seeds with shortness? So he took two types of parents. He named them as a P1 and P2 parents. P1 parents were having round seeds and tall height, and P2 parent were having wrinkled seeds and short height. He performed a cross-pollination between them, and cross-pollination produced the seeds. He saw the seed and got the F1 generation. So all the plants of the F1 generation were tall and had round seeds. So he concluded that tallness and round seeds were dominant traits. So tall character was dominant over short character, and round seed character was dominant upon wrinkled seed character. But what happened when these F1 progeny were used to generate F2 progeny by self-pollination? So F2 generation produced 4 different types of plants. Tall with round seeds, tall with wrinkled seeds, dwarf with round seeds, and dwarf with wrinkled seeds. But their number was not equal. Mendel observed about 1600 plants. He observed 900 were tall with round seeds. 300 were tall with wrinkled seeds. Next 300 were dwarf with round seeds. And remaining 100 were dwarf with wrinkled seeds. So he said that dihybrid ratio of F2 generation is 9:3:1. So Mendel explained this ratio by his independent assortment rule. He said that any factor can be combined with any other factor. So that means the tall plant can have round seeds as well as it can have wrinkled seed also. Likewise, the short plant can have round seeds as well as wrinkled seeds. Thus, the tall or short trait and the round seed or wrinkled seed trait are independently inherited. But how do these traits get expressed? What is the mechanism of heredity? How does heredity work? Mendel had no idea about it because in that time no information was available about nucleus, about genes, about chromosomes. But now we know that it is a DNA that is the hereditary material. So a part of DNA that provides information for one protein is called the gene for that protein. It is the protein that controls a character. That means the tallness is because of the protein. The dwarfness is also because of protein. The round and wrinkled seeds are because of the proteins. The proteins may work as enzymes. So the plant height can thus depend on the amount of a particular plant hormone. In tall plants, the protein of a gene is functional and that produces more amount of plant hormones, which causes tallness in the plants. The amount of the plant hormone made will depend on the efficiency of the process for making it. In dwarf plants, the plant hormones which causes plant's height is in low amount because the enzyme and proteins are non-functional. Thus, the plant will be short. So that means we can say that genes control characteristics or traits. We already know that both parents contributes equally to the DNA of the progeny. That means both parents contributes a copy of the same gene. This means that we receive one copy of the same gene from one parent and the other copy of the same gene from other parent. This means a parent has two copies of the same gene and offspring also has the two copy of the same gene, one from father and one from its mother. And when offspring grows and becomes adult and starts sexual reproduction, then it gives one copy of a gene to its offspring. And the offspring receives second copy of the same gene from the other parent. By this way, we can conclude that we all are having two copies of the same gene. This condition of genes is known as diploid. This means that each pea plant must have two sets of all genes, one inherited from each parent. For this mechanism to work, each germ cell must have only one gene set. So germ cells are haploid. Germ cells are produced by the process of meiosis. So now we are able to understand that how do germ cells make a single set of genes from the normal two copies that all other cells in the body have. But have you ever thought what is the location of genes? Genes are present on chromosomes. That means thus each cell will have two copies of each chromosome, one each from the male and female parents. Every germ cell will take one chromosome from each pair, and these may be of either maternal or paternal origin. When two germ cells combine, they will restore the normal number of chromosomes in the progeny, ensuring the stability of the DNA of the species. Such a mechanism of inheritance explains the results of the Mendel experiments and is used by all sexually reproducing organisms. But asexually reproducing organisms also follow similar rules of inheritance. So now we are in the sex determination. We know that two sexes, means female and male, participates in sexual reproduction, and they are quite different from each other for a number of reasons. So we can easily distinguish between male and female. That means there is variation in female and male. And the variation is because of different types of genes. So few genes are present in males only and few genes are present in females only. We already know that genes are present on chromosomes. So the gene which causes maleness is called as Y chromosome. So the maleness is because of X and Y chromosome. And the femaleness is because of XX chromosome. Human has 46 chromosomes. Out of these 46, 44 chromosomes are known as autosomes. These 44 chromosomes are known as 22 pairs of chromosomes, and the remaining chromosomes are called as sex chromosomes. When 22X gamete is fused with 22X of other gamete, a zygote is formed which is 44 plus XX, and this type of zygote is female. And when the sperm carries 22 plus Y chromosome fused with egg which is 22 plus X, it becomes a zygote which is 44 plus XY, and this zygote becomes a male. So most human chromosomes have a maternal and paternal copy, and we have 22 such pairs. But one pair called the sex chromosomes is odd in not always being a perfect pair. Women have a perfect pair of sex chromosomes, both called X. But men have a mismatched pair in which one is a normal sized X while the other is a short one called Y. So women are XX while men are XY. Half the children will be boys and half will be girls. All children will inherit an X chromosome from their mother regardless of whether they are boys or girls. Thus, the sex of the children will be determined by what they inherit from their father, not from mother. A child who inherits an X chromosome from her father will be a girl, and one who inherits a Y chromosome from him will be a boy.